My name is Rupal Mehta from Rush University, and I'm going to describe the JEM paper entitled Arachnoid Granulations are Lymphatic Conduits that Communicate with Bone Marrow and Dura-arachnoid Stroma. Arachnoid granulations are located at the brain surfaces, most prominently in perisagittal brain regions next to superior sagittal sinuses. However, they do not localize only to dural venous sinuses as defined in historical literature. Arachnoid granulations are unique to the central nervous system and are notably found across primates and many higher mammals. Here, on a schematic depiction, we see the general location of these structures in the brains of humans. In this study, we conducted systematic histologic and MRI analyses to evaluate frontal arachnoid granulation structure. Arachnoid granulations display apical dome and basal or neck regions. On histology and MRI exam, several arachnoid granulation types were discovered based on the location of dome embedment. In addition to intrasinus type, 4 non-sinus types were observed and included stromal, diploic, epidural, and subdural types. Prominent morphological heterogeneity was also noted with regard to size, lobation, cellular constitution, and degree and type of encapsulation. Using immunohistochemistry-based techniques, we interrogated the internal structure and discovered that granulations are composed of a collagenous framework. This internal core contained cavernous spaces that were populated by degenerated cells, degenerated cell elements, cytokines, and immune cells. Immunohistochemistry also showed that the granulation capsule was composed primarily by meningothelium. On high-resolution imaging, we identified perforations of the meningothelial capsule by variable caliber fissures, sinuses, and crypts. Furthermore, we found that a majority of granulations border on dura-arachnoid stroma adjacent to the superior sagittal sinus. This tissue contained arachnoidal and dural elements admixed with blood vessels, lymphatic vascular elements, degenerated cells and cell elements, cytokines, and immune cells, including T cells and MHC2 positive antigen presenting cells. These anatomical data imply that arachnoid granulations are fluid conduits as well as sites of antigen deposition, antigen processing, and immune activity. These findings have not previously been shown and may have broad implications as they suggest that arachnoid granulations have under-recognized roles in brain maintenance, homeostasis, and a range of diseases.